So chapter 8 and it's not, I mean, it's not really about the cells. It's not really chemistry. It's something in between, okay? So it's introduction to metabolism. And this part is quite essential, I think, for understanding what you're gonna deal with after the midterm, okay? So certain things might be quite familiar to you already. And there are a few concepts that I will ask you to pay attention. So let's start. So metabolism, I'm sure you heard that word, you operate this word many, many times. Can you tell me what you understand, like how you understand what is metabolism?
As you get older, metabolism slows down, and when you're younger, your metabolism is pretty fast. That's what I got from it.
Yes, but what is that? What is metabolism?
I don't really know.
You just know it's slow. It's fast when you're young and slows down.
Is it like a chemical process?
Yes.
That like maintains someone's body?
Absolutely. Yes, but metabolism is carried out a level of every single cell in your body. So every single cell in your body is actually does its part in terms of metabolism, and actually cells can be quite different in terms of it. For example, cells in the liver, they are very metabolic, very active. Cells, say like in your skin layer, I have much, much slower metabolism. But yes, metabolism is actually all biochemical processes that take place in organism, but also at the cellular level. Okay, and we'll skip. So we're gonna get to
So,
this.
Professor.
Yes.
Metabolism is basically all your cells in your body slowing down or speeding up.
I should say metabolism is all biochemical processes. Like I should say, all chemical reactions that happen in your cells.
Okay.
And in some cells, it can be slow. In some cells, it can be fast. I shouldn't say that, what people usually understand by slowing down metabolism is how fast your body actually processes food.
Real Dutch guys like you stand by and stroke your beard like a wise man instead of telling the president to get on it when you have power. How about tell your brother to get on it? My brother will stand for his own.
All right. Okay, so yeah, what we understand by metabolism is that you, like, when you get older, you don't burn calories as fast as you used to when you were younger, right? It's like it's much easier, for example, to control your weight when you're young, just by physical activities, right? But when you get older, it's getting harder and harder and harder. And metabolism, if you like think about level of like personal level, not on the level of cells, but level of the entire person, it's quite specific because huge role in metabolism play hormones, right? And the hormonal picture that you have when you're young and when you have it when you get older is quite different. Okay, so that's what you can actually, point the finger and say that's what changes. Okay, Brianna.
Okay, gotcha.
So metabolism. So totality of all organisms' chemical reactions, right? And it comes from, yes, interaction between the molecules on molecular level. And if we talk about metabolism, so there's something called metabolic pathways. So metabolic pathways is actually types of the reactions, like specific types of the reactions that happen within the cell, okay? And these types of reactions, they have a specific way how they are propagated. Like, for example, in the pathway, you start with a molecule and it might take like few steps to produce a result, an end result, okay? And in this pathway, it might actually take a number of steps, and each step has its own enzyme, which is a protein that actually uh, coordinates these chemical reactions. So you can see in pathway, right? So you start with A, you get to D, and these are the steps from A to B, from B to C, from C to D, and different enzymes can actually, they can make each step happen. I think good example could be, guys, you familiar with the Parkinson disease? Have you heard about Parkinson disease? No, just say yes, no.
Yes.
Yes. So
Yes.
what happens in Parkinson disease?
You have, I know it's, you're very slow in producing movement, certain types like walking, due to the lack of dopamine in the brain.
Wow, yes, absolutely. Yes, there is an area in the brain that produces like 80% of dopamine, and cells in this area die off for unknown reasons. And the best treatment could be is to supply dopamine as a medication, right, to alleviate symptoms. But the problem is that dopamine, in the way it is, won't be able to get into the nervous system because there's something called blood-brain barrier. Blood-brain barrier means that substances, molecules that could circulate around the body and get into the blood, out of the blood, can do the same in terms of brain. So brain is very selective in terms of which molecules can get from the blood into the cells. And what usually people who have Parkinson get, they get something called L-DOPA. L-DOPA is one of their steps, actually metabolic steps on the metabolic pathway to prepare dopamine. L-DOPA is a small molecule and it's more like hydrophobic, so it can actually get into the brain and get included into the pathway for synthesis of dopamine, okay? So sometimes when you can't supply the end product as a medication, you can supply one of their like intermediate products of the metabolic pathway, okay? Okay, guys, unfortunately, the problem with L-DOPA, it doesn't work for a long period of time, so it can cause elevation of the symptoms, but eventually it stops working for also unknown reasons. And please, two definitions: catabolic pathways. So catabolic pathways, they break down complex molecules to simpler ones, right? Like in respiration, glucose in presence of oxygen goes down to carbon dioxide, water, and releases energy. So breaking down, whenever you break things down, energy is released. Anabolic pathways, opposite. They build molecules from simple ones, and usually to build something, energy is required, right? Like energy is required to make proteins from amino acids, right? So catabolic breaking down complex to simple, energy is released. Anabolic, from simple to complex, energy is consumed. And sometimes catabolic, they also describe as downhill, and anabolic, uphill. Okay, downhill means breaking down, and anabolic, building up. Okay. And bioenergetics is actually a part of study of, I should say it's a part of biochemistry, is just to figure out how energy is released or consumed or exchanged during metabolism. Okay, so a few concepts that I hope you remember them. So what is energy? First of all, energy is capacity to do work or to cause change, okay? And there sometimes, energy has to overcome certain what's called opposing forces, which could be gravity and friction, right? Gravity, whenever everything is, is pulled down, and friction is usually resistance, like mechanical resistance. And energy can be kinetic, right? So kinetic is energy associated with emotion. A good example of kinetic energy is a mill, right? It could be water mill, it could be windmill. But their energy of the motion could be used, for example, for other type of work, like a water mill could be used to churn wheat to make a flour, right? Or you see water going through the dam turns turbines, right? Like in hydroelectric stations. So energy of motion. And I should say, like, that's something that amazes me in kids, right? So like by the end of the day, you see 6, 7, 8 years old, they are like in constant motion, which is like wow, how by the end of the day you can still jump. So I always think like there's like so much power in these kids that you can convert it into electricity. You can convert this energy into like so many things. Yes, guys. What do you think? Why do they have so much energy comparing to adults?
Because they're young.
Mm-hmm. Yes, the metabolism is fast. They should burn everything, right? Especially, you see, my 7 years old, he hardly eats anything. My gosh, but he is like, I don't know, he's in constant motion. But I think it's in these days it's different because these poor kids, they stay home for school and they don't have a chance, to run during the breaks. Okay, thermal energy. So thermal energy is also kinetic energy. And this is energy when heat is transferred, right, from one object to another. It's like you're warming your hands with a, cup of hot tea or coffee. Heat energy, again, can radiate from like hot objects, but there's also heat energy that comes from sun, which can also be captured for sun energy by solar panels. And this energy is also used to run photosynthesis. We're going to talk about photosynthesis, and you'll learn more actually how plants tap on this solar energy. Potential energy. So potential energy is a form of energy that it's kind of like you said, like it's locked. It's locked. It's not doing work yet, but it is able to do so. I think example could be battery, right? So batteries, before you put them into, let's say, flashlight, right? They have potential energy. Molecules, like molecules of glucose, molecules of fatty acids, fats. They also have potential energy that can be released, right, during the right biochemical process. Or you can say a bowl on the top of their shelf. No, I should say maybe like, yeah, let's say bowl on the top of the shelf before it falls and hits you on the head also has a potential energy, right? And the energy of blow is actually how this potential energy is translated, okay? Chemical energy, and that's what I was talking about, is energy available for chemical reactions. So one more time, I think I told you, people like sweets because once they look at sweets, they see energy, right? Potential energy in sucrose, potential energy in starch that eventually will be broken down to glucose, and glucose will provide your cells with ATP. Second, guys, anything, like any questions? Any questions, guys? I've just hear some noise and I'm not sure what is that. Am I too fast? Should I slow down?
No, you're good, Professor.
Okay. So far it's clear about kinetic, thermal, chemical, and potential energy, right? And energy can be converted, I'm pretty sure you convert it from one form to another, right? Like potential energy locked in the batteries, you push a button and a fan is a pure kinetic energy, right? The motion. Energy from food, chemical energy from food allows our cells to operate, allows our muscles to contract so we can move, right? Thermal energy could be converted to mechanical energy, like in all the steam engine trains, right? So heat, steam, and movement, motion. Yeah, so like this diagram, right? Diver has more potential energy on the platform than in the water, right? Jumps, potential energy to kinetic energy. And now less potential energy in the water. And climbing is kinetic energy of the muscle movement to potential energy again, right? So it doesn't require much energy to jump, but it will take more energy to climb. And let me share the sound. 1 second.
Energy is the capacity to cause change. Kinetic energy is the energy associated with motion. Energy that is not kinetic is called potential energy and is energy that matter possesses because of its location or structure. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy. Now let us see an example of how energy can be conserved. The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow. When the arrow hits the target, its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let us rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, some of the energy can be used to do work, but some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. Watch the archer fire the arrow again. The red glow indicates heat. As the arrow flies through the air, Heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Heat energy is a very disordered kind of energy. It has the highest amount of entropy or disorder of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. When your body breaks down food molecules, the stored potential energy from food can be converted to kinetic energy. Look at the archer's arm to see how energy is converted in a muscle. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP. Water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work, such as pulling back a bowstring, but the heat energy that is generated cannot be used to do work.
Okay. So in this video, heat was mentioned, right? So heat is definitely, it's energy on its own. And one of the reasons that, like, you can say, like, where does the energy go? Like, where the energy from the muscles go while you get eventually tired. And you can say most of the energy, yes, is heat generated by muscular activity, right? And that's one of the reasons why we're sweating. And thermodynamics is actually approach to understand, like, what's going on with energy related to heat, right? And thermodynamics, yes, study of energy transformation in a collection of matter. And when you talk about isolated system is like a system that is a kind of not really affected much by events outside, and without much fluctuations in terms of temperature and pressure, right? So this is called an isolated system. And it gives example, liquid in the thermos bottle will be unable to exchange energy or matter with the surroundings. So that's how the heat is preserved. It's preserved by a special material in the thermos. But if you put hot water in the cup, right, over time, it's gonna cool down, right? So this heat will radiate into the air. So open system, not enclosed system. So heat could be transferred easily. Like, as I said, you pour hot water in the cup. And our organisms, they considered open system because we constantly exchange energy with their environment, right? And we radiate heat all the time. Just, you know, as I said, muscular activity generates a lot of heat. Okay, so I think we're gonna talk about only two laws of thermodynamics. They are quite simple, I should say. And the most important law is energy can be transferred and transformed, but it can be created or destroyed, right? So, and question like, where does most of the energy go if it can't be destroyed? Yes, I'm saying it takes a form of heat, okay? It's also called the principle of conservation of energy. So we can, for example, calculate how much calories, like the value of calories from our food by, for example, measuring amount of carbon dioxide generated through breathing, by measuring a change, a fluctuation in the temperature. So I think there are ways how to measure energy value of the food in relation to our metabolism, right? So how it's transformed. But the thing is that it can't appear from nothing and it doesn't disappear. It just takes different forms. Okay, so this is the first law of thermodynamics. Therma is heat and dynamics means in motion. Okay, so we see chemical energy of food is transformed to kinetic energy of motion, and eventually it's released as a heat. Okay. So second law of thermodynamics is actually every energy transfer or transformation increases entropy of the universe. So what is entropy? When we talked about diffusion last week, remember I told you molecules are in motion, constant motion. Fast in gases, maybe a bit slow in the liquids, but depends on the temperature. But it also exists in solids, right? So different rate, but there's still motion. How they move, which direction they move. They can move to from down concentration gradient, right? But if they get energy, say for example, heat energy, they tend to move faster, right? And interestingly, in nature, this movement doesn't have any order, right? I mean, yes, from high pressure to low pressure, from high concentration to low concentration, yes, but if you take many different systems, you won't find like universal law of direction, right? So entropy is a term that measures this molecular disorder, right? Like, again, imagine you heat water, right? So you increase the movement of the molecules, you increase more and more and more as the temperature goes up, and this water starts to boil. But at this point, you can't really say which way molecules go, right? So there's pretty much like randomness of the movement. So this is their entropy. Okay, and interestingly, depending on their energy, right, provided, entropy can increase and entropy can decrease. Any questions, guys? Are you with me?
No questions, Professor.
Seriously? No questions? Yes.
It's pretty much straightforward.
Awesome. Awesome, guys. It's, how to say, I think I told you that chemistry, I had to have a name because my mom, she was a chemistry teacher, right? Physics, I was like really, really unlucky in the sense it took actually for me to finish my bachelor's to like get a better understanding of physics. And when I was looking at this thermodynamics, entropy, whatever, it's like, wow. Okay. So, yes, and we definitely increase disorder. We increase the entropy through our metabolism, right? We increase it a lot depending on what we do. And guys, there are like few more things, and it's gonna get like it's gonna have like more solid ground. So now this slide basically says that whatever increases entropy can happen spontaneously, right? So spontaneous process can happen without any particular energy input, and it can happen quickly. It can take long time. Like a good example could be radioactive decay, right? Like uranium that decays, I should say, fairly fast, right? But if you take another molecule, for example, you take isotope of carbon, the same spontaneous decay happens much slower, right? Remember, decay is when isotopes, they release subparticles from the neutrons. Like slow spontaneous process could be rusting of iron, right? But I don't know, nothing better comes to my mind than like explosion of corpses, which is also a spontaneous process. I don't know why. Okay, and order and disorder. So we talked at the beginning that in biology there is order, from atom to molecule, from molecule to macromolecule, and from macromolecules to cells, tissues, right? And it's true and it's not true, right? So it's true in terms of organization, like structural organization, but in terms of energy, it's not exactly true, because again, you have metabolic pathways going on all the time, right? And during these metabolic pathways, you might have a wide variety of energy flows, right? This is order, right? So glass sponge, and this is Sagrada Familia in Barcelona, which looks kind of similar to this glass sponge. And so again, while you have structural order, right, you can say there's quite a disorder in terms of energy flows, okay? So whatever happens. But just remember, eventually, energy by the end of the road is converted to heat, right? And the heat, whenever is produced, it increases the entropy. Okay. Now, I think by the end of 19th century, there was a guy, he was a professor, I think of Yale University, I think, or Columbia. His last name was Gibbs. So Gibbs was talking about something called free energy. So what is free energy? Is energy that could be either facilitate spontaneous reaction, right? Or if its energy will be consumed, right? So consumed in order to carry out chemical reaction. And he said that free energy, yes, is the portion of system energy, and he's talking about enclosed system, that can do work when temperature and pressure are pretty much stable, right? So stable, uniform, and for example, on the level of a cell, so free energy. There's a formula. If you don't like formulas, don't worry, it's okay. So it's not a hard one, right? So free energy, again, closed system. And it's related to temperature change, and it's related to enthalpy and entropy. So first of all, what is enthalpy? Enthalpy is like a total energy available in the system. Entropy, we just talked about like disorder, rate of disorder. T is temperature measured in Kelvin, right? In delta is a symbol that usually represents a change, right? Not like a, it's not like a particular value, but it's a change in their value, right? So change in energy. So delta G is change in free energy, which equals change in total energy minus change in entropy times like stable temperature, okay? So you can calculate this value of change in free energy for many systems. And interestingly, when this change in energy is negative, it's usually related to spontaneous processes. When it's zero, it's or positive, it's for non-spontaneous processes where energy has to be consumed, okay? I'm just thinking about like a good example. So this is like diffusion. This is the chemical reaction. Like again, spontaneous processes means they release energy, right? So it's a negativity, like release. Non-spontaneous processes, they are either zero or positive because they consume energy, okay? So if you imagine that, like, what do you think? Burning is spontaneous or non-spontaneous process?
Spontaneous?
What?
Spontaneous?
Burning? No, it's non-spontaneous actually. But I should say like glowing, for example, will be definitely a spontaneous process, right? Energy is released, but burning, that's like a conversion of energy. Probably it's not a good example. But anyways, okay, so the most important concept from this one is negativity or positivity. Releasing energy, consuming energy. And as I said, delta, this is the symbol, is difference, right? It's a change. So final state minus initial state, right? Just 1 second, guys. It's 9:34. I just have to move to a different room. So do you want to take like a brief break till, let's say, 9:43? 10 minutes?
Sure.
Okay, guys. So I'll see you in 10 minutes.
Great.
You know what? I was thinking about like a good analogy of this, what's like free energy. And I think a really good analogy could be a balloon, right? So you want to inflate a balloon, right? So you apply energy, like blowing air into balloon. So in this case, the change will be positive, right? Energy is applied. Now you release this balloon, and it's going to be spontaneous reaction, right? You just release it, and the air will flow out, and this change will be negative. Okay, so negative, system loses free energy. And the thing is that whenever system accumulated the energy, right, and is losing it, it contributes to stability of the system. And that's what actually most of their processes aim for. They aim for stability. And I think this is a nice diagram, right? So jumping into the water, right? So standing here, potential energy, right? Potential energy, but less stable. Jumping into the water, right? You release the energy, less energy, and you are more stable. And you can also contribute this change in energy to gravitational force, right? Gravitational motion. Less stable, release energy, more stable. Diffusion, the same process, right? Moving from high concentration to low towards equilibrium stability. Chemical energy is locked in the molecule, right? You release it, you release this energy. Again, the system becomes more stable, okay? But once it becomes more stable, there is less work capacity because energy was already released, right? Somehow you have to pick up more energy for something else, okay? And you see, systems never move away from equilibrium spontaneously, right? Because to move away from equilibrium, you actually have to get energy, right? So you can say you release energy to get to equilibrium, but to move from equilibrium, energy has to be consumed. Okay, and two important concepts that you're actually going to see on your midterm. Exergonic reactions. Exergonic, outward, energy is released, right? Like breaking down glucose. Endergonic reactions, absorbs. So we can say catabolic processes, they are exergonic, because they release energy. Again, whenever you break anything, you release. Whenever you have to build something, energy has to be consumed. So endergonic will be for anabolic processes. Okay, now the question is like, how do you get energy for anabolic processes? Usually what you do what's called a coupling. Coupling, did I say it right? Coupling. Guys, how you say it? Coupling?
Wait, what's the word?
Like coupling. I can write it. Let me just write it. I'm not sure if I'm saying it right.
Oh, coupling.
Coupling, right? Coupling means like you bind, like you combine. Yeah, so the whole thing is that if you need energy, you can actually pick it up from exergonic reactions, right? So that's where the coupling comes from. You can use energy when something breaks in order to build something, right? So exergonic, right? So energy locked, released, and you get the products. Reactants, products. Now for endergonic, this is the change in the energy, right? This is the difference. So low energy is consumed and you produce products. Any questions so far, guys?
No, Professor.
No, not so far.
Okay, and where the energy comes from during this coupling. Coupling, when energy is released, it could be locked as a molecule of ATP, right? And this ATP molecules could be used for endergonic reactions, right? So let's watch this one.
Cells contain many different molecules that can engage in a variety of chemical reactions. When molecules react, for example when they collide and exchange parts, their atoms and bonds are rearranged. Reactants are rearranged to form products. Reactant and product molecules store potential energy in the arrangements of their atoms and bonds. Chemical reactions involve changes in bonding and changes in energy. We can plot potential energy on a graph and compare the potential energy of reactants and products. Using these molecules as an example, we can imagine two chemical reactions. Molecules AB and CD react to form molecules AC and BD, or vice versa. Among the two reactions shown, the reaction shown in the left graph has products with less potential energy than the reactants. In this reaction, energy is released to the surroundings as the reactants are rearranged to form products. The potential energy of the molecules drops. In the other reaction, the product molecules have more potential energy than the reactants. The reactants must absorb energy from their surroundings for this reaction to occur. Chemical reactions that release energy are called exergonic reactions. You might call exergonic reactions downhill reactions. Other reactions called endergonic reactions are uphill changes. They absorb energy from their surroundings. Exergonic reactions occur spontaneously, or that is, without a net addition of energy. The potential energy of the molecules decreases. It is easier for a cell to carry out a reaction that does not need additional energy input. A downhill change is easier than an uphill change. Endergonic reactions do not occur spontaneously. It is harder for a cell to carry out a reaction that needs additional energy input. An uphill change is harder than a downhill change.
Okay. So this is like a summary, and so magnitude of change in free energy is pretty much like how much, like the maximum amount of work the reaction can perform. Like if you measure this in calories or kilocalories, right? So one molecule, basically 1 mole of glucose during cellular respiration can release 686 kilocalories of energy, which is available for work, right? And so products by the end of the reaction means they have less free energy, right? The glucose itself had a lot, but the products already have less because the reaction was completed, right? And energy was released. And I also told you, whenever you break bonds, it's, yes, breaking bonds is usually, yes, you can also have to apply some energy to break something, but eventually you get energy released, more energy released, right? And again, by the end of the road, whenever you remember, system always tries to reach the balance. So once it reaches the balance, there is less free energy available in the system, right? And endergonic. So in endergonic reaction, you have to
apply energy for it to happen, but this energy will be stored in the product, right? So exergonic reaction, real energy is released, but products don't store energy anymore. In endergonic, energy is consumed and stored in the product. And the change in free energy is positive, so this reaction's definitely non-spontaneous because energy has to be applied. So if you take a look again at this diagram, right? Exergonic energy, reactants beginning, a lot of free energy is released, right? Products have less free energy. In endergonic, reactants have very low free energy. Energy is consumed, but products have a lot. It's like, you see, photosynthesis. Photosynthesis probably is a very good example. So you take carbon dioxide, you take water, right? And the energy is the sunlight energy, right? So this sunlight energy will help to convert carbon dioxide, water into glucose that will have high free energy. So high free energy of glucose will be released during respiration, right? Energy will be released and the end products, carbon dioxide and water, will have very low free energy. Guys, are you with me?
Yes.
Yes.
You sure? Okay. So this is the illustration actually of this flow of energy in the systems, right? So free energy is the change in free energy is less than zero, right? Negative. Negative, water easily flows, right? And then zero, equilibrium is reached, right? But what happens that in living organism, equilibrium is never reached. If you get a full equilibrium, zero energy, the cell will die, right? So this constant energy flow, which reflects metabolic pathways, as you see, that's what defines life, right? If everything stops, it's pretty much over, right? So there's constantly, something is going on, like one way or another, either metabolism or catabolism. Yeah, so this is the open hydroelectric system. That's what I just showed you. And this is like multi-step. So downhill, right? So downhill energy is released and you actually gonna see this very, very much nice illustration in, for example, respiration. Okay. So you see, this is like a multi-step system and that's what happens in cellular respiration. So one step after another, one after another, which is also, remember, metabolic pathway, right? But again, equilibrium is never reached. And the next part, we're going to talk about the ATP. I think a few slides will be about ATP. So it's a very interesting molecule. I'm quite curious myself, like where did this molecule come from? At what part of, you know, biological evolution it's actually appeared and was actually involved in powering metabolic processes in the cells, right? So, but let's take a look. So cell does different kinds of work, but here are the three main. So pushing endergonic reactions. anabolic processes. Transparent, right? Moving molecules across the membrane against the concentration gradient. And mechanical work, muscle contractions, right? Moving of cilia or flagella. So all these processes require fuel, right? We usually think about fuel as a glucose, and it's true. But just remember, one molecule of glucose through cellular respiration yields 30 or 32 ATP molecules. So eventually glucose is translated to ATP, and ATP is translated into fuel. And that's what I was talking about with the help of Jamarius. Energy coupling is when you use exergonic process to run the endergonic, right? And that's the ATP, right? Exergonic produce ATP, endergonic consume ATP.
Every working cell has to carry out thousands of endergonic reactions, reactions in which products have more potential energy than reactants. By definition, an endergonic reaction does not occur spontaneously. Then, what can a cell do to make an endergonic reaction happen? The trick is energy coupling, obtaining chemical energy from an exergonic reaction and then using the energy to drive an endergonic reaction. ATP is the key to energy coupling in the cell. In an exergonic reaction, ATP becomes ADP as a phosphate is removed. A reactant molecule acquires the phosphate from ATP and gains energy. Now, the reaction can proceed.
Okay, so ATP stands for adenosine triphosphate, and it's made of ribose. This is the ribose, which is our pentose. Pentose, you see the pentagon, right? 5 carbons, pentose. The same ribose actually is present in RNA. Adenine, which is also nitrogenous base, right? Adenine, like part of DNA. And there are 3 phosphates. So phosphate group is phosphorus, and each phosphorus has 4 oxygens attached. Now, pay attention. The key to ATP molecule, to understanding ATP molecule, why it locks so much energy, is that they all negatively charged. So being like all 3 negatively charged, it's like a spring, like a metallic spring that you press, right? Think about piece of paper that you fold, right? Like a spring. And think about metal spring. Which one will have more energy once you release? Tell me.
The metal spring?
Of course, the metal spring, right? So yes, so these negative charges, they pretty much lock the energy that is released when ATP undergoes dephosphorylation, okay? So dephosphorylation is actually release of one of their phosphate groups. So going from ATP to ADP. T stands for triphosphate, D stands for diphosphate, right? So ATP plus water will give you ADP plus phosphate. Okay, so let's take a look at this one.
The structure of ATP. ATP powers nearly all forms of cellular work by providing energy stored in its chemical bonds. It is composed of 3 main components: a 5-carbon ribose sugar molecule, adenine, a nitrogenous base, and a string of phosphate groups. Now let's construct an ATP molecule. Notice how the sugar ribose attaches to a nitrogenous base adenine, which is also bonded to a chain of 3 phosphate groups, the triphosphate group. During cellular respiration, glucose releases energy. This energy is stored when a phosphate group is added to ADP, adenosine diphosphate, forming ATP or adenosine triphosphate. ATP releases energy when the covalent bond between phosphate groups breaks during hydrolysis. This energy is used to drive other biochemical reactions.
So basically breaking ATP to ADP, like releasing one of the phosphates, that's what the energy that could be used for building new molecules. So this is like the space frame model of ATP, this is stick model of ATP, and that's what I was talking about. ATP plus water, right? Phosphate is released and energy is released as well. This is hydrolysis. Hydrolysis actually taking water in, okay? When you take water out and you add phosphate, you actually go opposite direction from ADP to ATP. Interestingly, what happens, energy is released from ATP to ADP. Then you have a bunch of ADPs. Energy could be used to go opposite direction, right? So cells actually run opposite reactions whenever energy is available to kind of like accumulate fuel, right? Just have a picture. Yes, this is like regeneration of ATP, so we're gonna get to this. Actually, I'm a little bit ahead of time. Okay, so that's I just explained to you that three negatively charged phosphate get a lot of potential energy. This molecule is unstable. Unstable means releases energy. And again, most of the cellular activity are provided by ATP. And phosphorylation is, again, phosphorylation when phosphate group from ATP could be transferred to other molecules. Okay. And again, this phosphorus could be used for many purposes. For example, when glucose enters the cells, right, it will be immediately phosphorylated. This phosphate group will be transferred to glucose, and the glucose will be locked inside the cell. Once glucose is locked inside the cell, it can undergo cellular respiration and release more energy that could be converted to ATP. So, or it could be used for some chemical reactions. For example, glucose plus ammonia produces molecule called glutamine, okay? But ATP actually participates in the process. So glutamic acid will get phosphorylated by ATP. So it's glutamic acid plus ATP, phosphate transferred to glutamic acid, right? Then it binds ammonia and you get glutamine, okay? So what basically happens, energy was applied, right? And energy is released. So kind of like balanced reaction, both ways. And transport, right? So transport work, we talked about sodium potassium pumps, right? That operated by ATP. And motion. For example, here you have a part of the cytoskeleton, right? This is the vesicle. Vesicle moves along their track on the cytoskeleton with the help of motor protein. So it literally kind of like walks along, but to walk, like our muscles need energy. So the same idea here, the walking is operated by ATP. I can also tell you that muscle contraction, like literally, like for your muscle to, just move a finger, your muscles will consume a bunch of ATP molecules. just for the muscle cell to contract or shorten. And that I was talking about regeneration. So regeneration of ATP, usually energy has to be applied to for the ATP to bind phosphate and become ATP again. There are different ways how cells do that, but as I said, cells usually, whenever they have free energy, they will phosphorylate ATP. I don't know, people who go to gym, they probably heard about creatine. Creatinine, guys, have you heard about that? I think it's one of their how you call it? How you call this? Like dietary supplements. Have you heard about that? The thing is that when your muscles move, right? So when you work out, your muscles break down a lot of ATPs, right? Cellular respiration requires oxygen. And if you're out of breath, if you're really, really tired, you kind of like, your cells kind of like run out of oxygen supply. And they start using other sources. And one of the sources is actually a creatine phosphate molecule. And this molecule is present in the muscles. And this is the molecule that just delivers phosphate to ADP. Okay. So, I mean, molecule that kind of like a phosphate carrier, so ATP could be regenerated. Are you with me? Quiet class.
Yes, no, Professor.
Quiet
Yeah.
people. Okay, thank you. So let's take a look at this one.
As organisms live and grow, they're constantly in the process of making and breaking bonds in molecules. Metabolism is the sum of all the chemical reactions that take place in an organism. Metabolism includes catabolism and anabolism. Catabolism includes the processes that break down complex molecules into simpler molecules while harvesting their energy and storing it, usually in the form of ATP. Anabolism includes the processes that build more complex molecules from simpler molecules. The energy acquired through catabolic processes is used to drive anabolic processes. Neither catabolism nor anabolism is completely efficient, so at each step, some of the available energy is lost into the environment as heat.
Okay. So the last part of the class actually. So last part of the class, like what mediates all this energy flow and mediates all these reactions. Actually, the answer is enzymes. Enzymes, remember, they're proteins which act as catalysts. So catalysts, they, I shouldn't say like speed up. I should say they modulate, they facilitate, they control, they basically control chemical reactions, but they're not consumed by reactions. So their presence actually can, their presence can actually make a difference, okay? So this way. And like, for example, sugar, sucrose. Sucrose, you can, which is a disaccharide, right? It's made of glucose and fructose. You can leave sucrose with water, right? For a long time and nothing gonna happen. It's not gonna break down to glucose and fructose on its own, even at the right conditions. But if you add enzyme, the sucrase, right? In a few seconds, it will be broken down to glucose and fructose, okay? So they really make a difference in terms of conducting chemical reactions. So sucrose plus water. And as I said, you just put sugar in the water and nothing gonna happen. But add sucrase, right? And you're gonna get glucose and fructose right away. And what is it all about? It's all about something called activation energy. So you have reactants and you have products, right? So reactants have to change, right? They have to change in order to be converted to products. It's like, I think your book gives a very good example. It's like when you want to put a key on their, on this like metal ring, right? You have to bend this metal ring to push the key through, right? So bending requires energy. So this is called activation energy. Activation energy, energy that it's needed for change in order to run chemical reaction, okay? And quite often, you know, for these reactions to happen spontaneously, the barrier could be way too high, right? So again, to make this happen, that's basically what a catalysts are for, right? So you see, initial energy needed to break the bonds is called the activation energy, the energy needed for transformation. Heat is usually can be supplied, right? Can supply activation energy. Can be unstable, and this can facilitate, but still, sometimes just heat is not enough. Okay, so you see, for some reactions, you see the activation energy is low enough. So thermal energy is sufficient to pass the barrier. But for most reactions, the activational energy is too high and you need a catalyst, okay? So one more time. Activational energy could be, can use heat, right? For some of the reactions to happen. But for some of them, again, the barrier that they have to overcome could be way too high. To help to overcome this barrier, that's what catalysts are for. So catalysts can speed up reaction. I should say they are really speeding up. They really make it more likely to happen, okay? So what catalysts do, they lower the activation energy barrier, right? So they can see without enzyme is way too high. With enzymes, it could be lowered, right? Once it's lowered, reaction is more likely to happen. Any questions at this point? I hope I didn't confuse you with their
No, Professor.
activation energy. Thank you, Dylan.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
So basically enzymes are, again, they are proteins that pretty much in charge of all metabolic processes in the cells, okay? So in case, when you talk about enzymes, reactions mediated by enzymes, the reactants are called a substrate. So substrate, they're like what you start with. So enzyme is very specific to substrate, okay? So one enzyme is usually for one particular reaction. They are not, they are, how to say, they're not capable of conducting different types, usually for one particular chemical reaction. And it's due to high specificity, right? Yes, so for example, this is an enzyme, and it's a fairly large protein, folded, like 3D, 4D shape. So this is the active site that substrate binds, right? Once substrate binds, enzyme usually changes its shape or conformation. Conformation is, again, the 3D shape. And once the product is available, enzyme usually goes back to its original form, okay? So you have substrate binds to enzyme, forms enzyme substrate complex. And enzymes usually end with ase, like sucrase, lactase, lipase, what else? Pepsin is not, but pepsin is not, pepsin is an enzyme, but it's not ended with ase. Amylase. And active site is, again, site where substrate binds. And it's usually, again, it's like a specific pocket or groove. And once it binds, as I said, it's usually enzyme will slightly change its shape. And it resembles like a handshake, right? Like two hands. Once you grab another hand for the handshake, right? They change the shape, right? So that's what induced fit, interaction between the different chemical groups on the substrate and on enzymes. So this is the induced fit, again, that resembles a handshake.
Watch how substrate binding to an enzyme active site induces a change in the tertiary structure of the enzyme. These images were created using X-ray diffraction data of hexokinase with and without bound glucose. Notice how glucose binding to hexokinase causes a conformational change in the enzyme that closes the active site. Credit: Mark B. Gerstein, Yale University.
Yes, so closing the active site once the substrate binds to enzyme. Okay, and so usually when substrate binds to enzyme, so there are like bonds are formed with the enzyme. It could be a hydrogen bond, it could be ionic bond, usually like very weak bonds. Okay, so enzyme will do the magic and then products are released, okay? And what you can see here, substrate binds to active site. Again, this binding to enzyme lowers the activational energy. They convert it to products. Products are released. And what you can see actually, you see the active site without products, without substrate, and the active site with substrate. So you can see that enzyme has to undergo conformational change in shape whenever substrates bind to.
Although every enzyme performs a different reaction on different substrates, they all go through the same general steps in accomplishing their reaction. These general steps include substrate binding, formation of the enzyme substrate complex, product formation and dissociation, and enzyme recovery. Like all catalysts, enzymes are unchanged by the overall reaction. Now let's look at each step in more detail. The first step in any enzymatic reaction is the binding of the substrate by the enzyme. The enzyme includes an active site that can accommodate the size, shape, and electron configuration of its substrate or substrates. According to the induced fit model of enzyme-substrate interaction, a perfect fit does not form until the enzyme and substrate bind to form an enzyme-substrate complex. After the substrate is bound in the active site, the enzyme-substrate complex is formed. The shape of the enzyme may change to allow for an efficient chemical reaction, and amino acids may accept or donate protons or electrons to facilitate the chemical reaction. Once the enzyme-substrate complex is formed and any reactive groups have moved to the right position, the reaction proceeds and products are formed. The products dissociate rapidly, leaving an empty active site. After the products leave the active site, the enzyme returns to its original configuration. Any groups that donated or accepted protons or electrons are also returned to their original state. Like all catalysts, the enzyme is unchanged by the overall reaction.
Okay, so that's the whole point. Substrate changes to products, but enzyme doesn't change on its own, right? So how they do so, right? How they lower activational energy. So the position of the substrates, right? So they have to orient the right parts of the molecules to each other so reaction can happen. They can be stretched to make bonds easier, like more breakable. It might actually provide a microenvironment that favors reaction, like in terms of pH. Amino acids in active site can participate in reaction. They can participate, but still, again, eventually enzyme stays the same after reaction. So rate of reaction is pretty much affected by availability of the enzyme and also by substrate concentration. If you have a lot of substrate molecules, enzyme could be saturated. Means saturated, it's actually all filled up, like all enzymes filled up, right? And it can slow down, but again, whenever you can manipulate concentration of substrate or concentration of enzyme, you can control the rate of the reactions. And local conditions. The most important conditions in terms of enzymatic activity, temperature and pH, okay? Temperature usually speeds up, right? Increases reaction rate at the beginning, but then once it's saturated, it can slow down. But also some proteins, they denaturate. They actually change their shape in high temperature. Like remember the eggs, right? So they go from like liquid to solid. So temperature usually has a very strong effect. Optimal temperature for most of the chemical reactions, in terms, again, most of the cells is the body temperature, but for some organisms, like it's called like thermophils, thermo temperature, feels like lovers. Certain archaea, they actually, their enzymatic activity can happen only at very high temperature, like in hot springs here. So thermophils, archaea, these are prokaryotes, so very special single-celled organisms that could be found in high temperature environments. Okay, so this is thermophilic, you see. For most, it's body temperature like 37 degrees Celsius, I'm sorry. But for thermophils, it could be 75 degrees Celsius that, you know, we can't really tolerate this temperature at all. And pH, so pH, it depends. Again, optimal pH will be from 6 to 8. But in our stomach, we have enzyme called pepsin. And while most of the proteins in our stomach, they usually can't tolerate this pH, it's optimal for pepsin that actually helps digest them. Literally like a scissor cuts the peptide bonds, breaking down proteins to amino acids. Okay, so trypsin, this is enzyme in intestine, which also actually breaks down peptide bonds. But trypsin works best at a little bit more alkalinic environment, while pepsin works the best at very low pH, like 2. Guys, are you with me? We have a little bit more.
Yes, Professor.
Okay. A little bit more. Maybe like another 10 minutes. Okay. So we just talked about enzymes, right? So cells have a variety of ways how to control enzymatic activity, right? So sometimes enzyme is not enough. So it should be not only optimal pH and temperature, but it needs additional molecules for reaction to be carried out. So it might need helpers, cofactors, non-protein helpers that usually have to bind to enzyme or maybe reversibly with a substrate. Inorganic cofactors usually could be iron, zinc, copper. That's why we need, by the way, we need minerals, because minerals could be like apart from their function, like calcium is a mineral, which is part of the bone matrix, but they also could be used as a cofactors, binding to enzymes, facilitating chemical reactions. Organic cofactors called coenzymes, vitamins, bunch of them, they are nothing more than coenzymes. And God forbid if they're missing, like missing, for example, vitamin C can result in very dangerous condition, disease called, anyone knows? Deficiency in vitamin C causes what disease?
I actually don't know. It's
Called scurvy. Scurvy, yeah, that's, oh my gosh, it's a very tough one. Deficiency in vitamin B6 causes horrible condition called pellagra. Pellagra is, wow, it's skin, intestinal problems. It can also cause dementia. And that's what like missing vitamins, which are, now you understand more, they are coenzymes. Now, usually there are two types of molecules that can affect enzymes. They could be activators. Activators activate enzymes like coenzymes or cofactors. But there are molecules that actually block their action, and they're called inhibitors. Inhibitors are blockers. And inhibitor can form bond with the enzyme, and once the bond is formed, it could be actually irreversible. So usually they don't form covalent bonds, but they form again weak bonds like hydrogen or ionic. So inhibitor could be competitive. What is competitive inhibitor? So you see enzyme and substrate active site. Competitive inhibitor competes with the substrate. It takes the substrate spot, and here you go. The enzyme basically shuts down, right? Because instead of binding the substrate, it's already blocked by competitive inhibitor. So if you increase concentration then of their substrate, actually substrate could compete with inhibitor and actually can overcome this problem. So this is competitive inhibitor.
A competitive inhibitor is a substance that is structurally similar to the enzyme substrate, but which does not react to form products. For example, sulfanilamide is a competitive inhibitor for the enzyme whose normal substrate is para-aminobenzoic acid, PABA. Note the similarity in molecular structure of PABA and sulfanilamide. In competitive inhibition, competitive inhibitors compete with the substrate molecules for the binding site, slowing catalysis. However, if the concentration of substrate is much higher than the concentration of competitive inhibitor, the substrate will outcompete the inhibitor for the enzyme active site, and catalysis will continue.
Okay, so competitive basically competes with the substrate. Non-competitive don't do anything with the substrate. What they do, they basically bind to enzyme and shut them down by usually changing their shape. Okay, so what you can see, non-competitive inhibitor by binding to enzyme changes the active site, so substrate can't bind. In this case, you can overcome inhibition by increasing loss of substrate because they can't do much. So it's like pushing a button, right, on the enzyme, turning it off by non-competitive inhibitor.
Non-competitive inhibitors bind to a location on the enzyme other than the active site. For example, the HIV medication nevirapine is a non-competitive inhibitor of HIV reverse transcriptase. Nevirapine does not bind to the reverse transcriptase active site. The binding of the inhibitor causes the enzyme to change shape, so the active site is no longer able to perform the reaction efficiently. Since the non-competitive inhibitor binds in a different binding site than the substrate, increasing the concentration of substrate has no effect on non-competitive inhibition.
Makes sense, right? So you see toxins, poisons, they are irreversible enzymes inhibitors, right? Like you see when this toxic gas was used during attack in Syria, it was sarin gas, and sarin binds to enzyme called acetylcholine esterase, which actually functions in the nervous system. It controls the activity of neurotransmitter called acetylcholine, right? And it's a very common enzyme, and if you shut it down, you basically kill the person. Pesticides, antibiotics, again, bacteria toxins, they quite often act as irreversible inhibitors. Now, evolution. So enzymes developed over time, and since enzymes are proteins, they're coded by DNA, right? So any change in the code can actually affect the shape of their enzyme. And eventually, I think like trials and errors, you know, this mutual relationship between enzymes and substrate developed through the change in genetic code over billions of years of evolution. Okay, and so yeah, there's like a few more things here that I would like to talk. Again, like imagine again, bunch of biochemical reactions that run inside a cell. This processes, they have to be coordinated, right? They're usually coordinated by genes. So, but genes, remember, they code for proteins. And these proteins basically, you know, they can act as enzymes, but they can also control activity of enzymes as well, right? So certain pathways could be turned on, certain pathways could be turned off. The best way to control is actually to change the conformational shape of the molecule. Like non-competitive inhibitor, remember, binds to enzyme and changes the shape, so enzymes become inactive. Allosteric regulation is basically binding of regulatory molecules, right? And these regulatory molecules, not necessarily inhibitors in this case, they can affect the shape and they can affect the function of enzyme, okay? So this is allosteric regulation, binding of the molecules. And quite often the molecule that binds can be the product itself. Okay, so you're gonna see. So activation and inhibition. You see enzymes usually made of subunits, polypeptide subunits that have their own shape, right? And this shape, again, is the binding shape for allosteric regulation. So protein already created with a site that could be used for regulation of its activity. And you see, they can change back and forth to active and to inactive site. So molecule can bind to this allosteric site and again can activate it or block it. So you see allosteric activators or inhibitors. So this is the allosteric enzyme made of 4 active sites, right? So this is the substrate and this is the activator, right? This is non-active site and inhibitor binds and inactive form stable, okay? So what you can see, it can go from active to inactive but being an active shape is not enough. It also needs a activator, okay? So active site, active shape plus activator, here you go. Inactive shape with inhibitor and it's blocked. So cooperativity is when a substrate binds to one active site, triggers a shape change enzyme that basically stabilizes its activity. So in this case, the substrate itself can become an activator. So in active form, and whenever substrate is able to bind to active form, it stabilizes its activity shape. But as I said, allosteric, it's really about changing of the shape of enzyme, right? So it's, I should say the word allosteric pretty much corresponds to conformational change in shape. And feedback inhibition is when basically enzyme has a binding site for the product, okay? It's a very common way of regulating synthesis of molecules, right? So cells have to know when to stop. The best way to know when to stop is once concentration of the product is reaching certain point, the product can bind to enzyme itself and block it. So no more is needed, right? Made enough, you can stop. So what you can see here is its enzyme that does, yes, thymine deaminase, which removes the amino group from their amino acid, right? So you can see this metabolic pathway. You get the end product called isoleucine, right? Isoleucine binds to enzyme and make it inactive, right? So one more time, feedback inhibition is to regulate rate of synthesis, right? When the end product can actually tell you to stop. And where you can find enzymes in the cell, like if you look at the mitochondria, you can find a bunch of them because that's you're going to see when we talk about their cellular respiration. You can find them. You can find them definitely in the nucleus. You can find them on endoplasmic reticulum. You can find them in lysosomes. So pretty much like you know, wherever you can see organelles in action, from protein synthesis to cellular respiration. Okay, I hope this material didn't confuse you. I hope so.